To understand the bottleneck in modern pharmacology, we have to look at the failure rate in clinical trials. 90% of drug candidates that enter human testing are ultimately abandoned. The primary biological roadblock is inaccurate initial binding affinity mapping, meaning a molecule that looks promising on paper fails to securely attach to its target protein in a complex biological environment. Because these docking failures are often discovered late in development, the traditional discovery pipeline stretches out to 10 or 12 years. Identifying poor candidates early is the only way to compress that timeline. Classical high-throughput screening attempts to solve this by testing massive chemical libraries. But simulating precise molecular dynamics for millions of compounds requires prohibitive hardware costs, making it unscalable for broad initial sweeps. Early machine learning models, specifically simple recurrent neural networks, tried to shortcut this calculation. They ingested one-dimensional sequence data, but consistently failed to accurately capture the complex 3D spatial representations required for true lock-and-key thermodynamic binding. A viable modern screening pipeline must navigate between these two extremes. It has to process candidates faster than classical high-throughput screening, while maintaining significantly higher accuracy than baseline sequential neural networks, directly reducing downstream clinical variance. The SUT Computer Engineering Technology team proposed a solution to this exact latency and accuracy trade-off. They call it Drug Target AI. The core architectural thesis of the platform is a hybrid, two-stage system designed to filter and verify sequentially. This diagram outlines the framework. Stage 1 is a predictive deep learning layer. It performs near-instantaneous abstract scoring of millions of candidates, acting as a rapid filter before the highest-scoring molecules feed into the second stage. That second block is Stage 2, the deterministic biophysical validation layer. Here, the system performs localized, strict physical mapping of only those top-tier candidates identified in the first stage. By isolating the heavy computational lifting to Stage 2, this dual-modality approach avoids running full biophysical simulations on the entire massive candidate pool, saving thousands of hours of processing time. Abstract predictive scores carry little weight without structural verification. An effective engineering solution must bridge fast heuristic screening with the strict laws of physical chemistry. To achieve that rapid inference in Stage 1, the underlying machine learning architecture relies on processing biology as a language. The processing pipeline handles the target protein by ingesting raw amino acid sequences, which are essentially strings of characters representing the biological structure. This flow illustrates the dual encoder setup. On the left, the protein sequence is fed into Hugging Face's ESM transformer. This model extracts a high-dimensional semantic representation of the protein's folded structure. Parallel to this, on the right side of the split screen, the system processes the drug candidate by ingesting its SMILE string, a text-based representation of its chemical structure. These SMILE strings are fed into the ChemBerta transformer, which encodes the molecular linguistics of the chemical compounds, mapping out their properties in mathematical space. The critical architectural juncture occurs when these two distinct outputs merge. The encoded representations from both ESM2 and ChemBerta are concatenated into a single shared multidimensional latent space. This unified dataset is then passed through final fully connected layers, which distill the complex interaction down to a singular numerical regression score, representing the predicted affinity. Standard one-dimensional convolutional neural networks struggle with this because they evaluate data locally. Transformers utilize attention mechanisms to accurately capture long-range biological dependencies across the entire sequence. By treating biology as a translatable language, the system completely bypasses the need to manually engineer complex physical features for initial screening. The primary constraint of a dual encoder setup is hardware. Fully fine-tuning massive models requires extreme amounts of GPU VRAM, making it impractical for standard servers. The targeted algorithmic solution to bypass these hardware limits is low-rank adaptation, or LoRa. As shown in this matrix decomposition diagram, LoRa works by freezing the massive pre-trained model weights. Instead of updating the entire dense data block, It injects small, trainable rank decomposition matrices directly into the transformer architecture. By only training these smaller matrices, the system drastically reduces the total number of trainable parameters. It achieves the necessary task-specific optimization while maintaining the structural fidelity of the original massive model. The efficiency of this training pipeline is validated by its empirical output. This table presents the final predictive benchmarks for the model. Drug Target AI achieved a root mean square error of 1.6276 and a mean squared error of 2.2167. That RMSC value specifically signifies the model's tight error margin when predicting the actual thermodynamic binding affinity, meaning the abstract score closely mirrors real-world chemical interactions. When contrasted against the higher error rates of the classical RNN and 1D CNN baselines, the quantitative performance delta proves the model's superiority in predictive accuracy. The LoRa-optimized dual transformer architecture effectively solves the accuracy and latency paradox defined in the initial hypothesis. Moving from machine learning theory to full-stack software engineering, we transition to the production-ready deployment stack built by Mohammed Ashraf. The application architecture consists of a Next.js client environment communicating asynchronously with a fast API Python backend designed to handle intensive parallel requests. A computational chemist begins by interacting with the predictive panel, inputting their target data into the interface. During the live inference pipeline, raw SMILE strings and UniProt IDs are routed to the backend models, processed instantly to return a dynamic regression score. However, there is a critical limitation at this stage. The regression score is mathematically sound, but it lacks spatial proof of a viable dock. A complete tool for computational chemists requires a transition from abstract numerical inference directly into verifiable 3D spatial geometry. To provide that spatial proof, the platform integrates WebGL and 3dmol.js viewers directly into the interface. Behind the scenes, the backend utilizes RDKit and Autodoc Vina version 1.2.5 to transition the highest-scoring one-dimensional candidates into a strict 3D coordinate space. The system performs a multi-pose docking sweep, computing the top 10 most stable thermodynamic conformations for the user to manually review. Because clinical research requires rigorous historical tracking and experiment replication, back-end data management is just as crucial as the visualizations. On the prediction history dashboard, affinity score and confidence columns quantify past inferences. Each row maps a drug SMILE string to its paired protein sequence. Finally, rerun affinity and rerun docking API triggers allow researchers to instantly replicate archived experiments locally. Drug Target AI operates as a complete production-ready ecosystem designed to systematically accelerate and deterministically verify the next generation of pharmacological screening.